Having understood about pollen pistil interaction, we have understood that there is this pollen and pistil, there is active dialogue between them. So if the pollen grain is not of right type, it can be rejected. So using this knowledge, there are plant breeders who are utilizing this knowledge, who are using this principle to bring the desired pollen grain in front of that, and this pollen grain which is of having the desired characters, they are bringing it to the stigma so that they are creating the hybrid varieties, they are creating the better varieties of better characters. So this is in case of artificial hybridization. So here there is a human intervention to create this pollination. So this pollination is between selective plants. They are selecting the plants which are of desired characters, and these desired character plants are brought together by this pollination approach. So this artificial hybridization, it is one of the major approach for the crop improvement program. So in the crop improvement program, they are creating the varieties, they are creating the hybrid varieties using this approach. So in this artificial hybridization is also one such approach to create the better varieties. So in this, what they are doing is they are bringing the desired pollen grain from the male parent to the female parent. So what do you mean by desired pollen grains? So this desired pollen grain, which is referring to having the desired characters. Like suppose if you say in a crop higher yielding is a desired character. So like if a plant is yielding more number of fruits, then it is a higher yielding variety. So that is a desired character there. Higher yield is a desired character. Likewise, it can be fruit shape, it can be fruit color, even it can be the fruit sweetness, or it can be number of flowers. So any character can be desired character based on the approach. So here there are some desired character present in the male and female. They are selected from the male and female plants. So those desired characters are brought together through this pollination approach. So this is the artificial hybridization. So desired pollen grains are taken up for this pollination. And meanwhile, what they do is they are protecting the stigma not to get contaminated. What do you mean by contaminated? So this contamination is referring to uh, the unwanted pollen grains. So when unwanted pollen grains are landing on the stigma, it is said to be contamination here. So contamination is referred to unwanted pollen grains. So in order to stop this contamination, there are a few techniques which are called as emasculation and bagging. So these are physical barrier techniques where they are usually stopping the pollen grain to the unwanted pollen grain to land on the stigma. So they are doing it by emasculation and bagging technique. So we are going to understand this in the bisexual flowers and unisexual flowers both. So first, starting with the bisexual flowers, we know in the bisexual flowers they are having both male and female parts together in the same flower. So in such case, if it is a female parent, what they have to do is they have to remove the male parent. So the anther should be removed from the female female parent of the flower. So here removal of anther from the bisexual flower. When should it be removed? It should be removed before anther dehiscence so that none of the pollen grain are landing on the stigma. So before anther is dehiscing, this anther should be removed from the bisexual flower so that there is no contamination occurring. So this step of removal of anther from the bisexual flower before its dehiscence is called as emasculation. So emasculation is seen in only bisexual flowers because anther and stigma, they are present together only in the bisexual flowers. In case of unisexual flowers, emasculation step is skipped. Okay, so next, moving on, the next step after emasculation, since there is a stigma which is free open, so in order to cover that, because there may be contamination from the unwanted pollen grain, so that stigma should be protected. In order to protect that, the breeders, they are taking the butter paper to prevent the contamination, and using that butter paper they are covering the stigma so that there is no contamination occurring in this. So this process of covering stigma using the butter paper is called as bagging. And bagging after reaching the receptive stage. So this stigma, after some time, it might reach the receptive stage. When we say receptive, it means that the stigma is able to take the pollen grains and allow these pollen grains to germinate the pollen tube and let its content to move further for the fertilization. So when the stigma of a bagged flower it is attaining the receptive stage, these mature pollen grains from the desired parent. So they are desired pollen grains which are brought from the desired parent, the desired male parent. They are bringing that pollen grain and they are dusted on the stigma. They are dusted on the stigma so that the pollination can occur and then fertilization can move further. And for these flowers are again they are rebagged. Why there is a need of rebag here? Because there may be another set of pollen grains which are unwanted might come again, and individually they might form the pollen tube and then let its content germinate, and then it will create the unwanted variety. So therefore it needs to be rebagged so that there is no contamination even after the pollination from the desired pollen grain. So there is a rebagging and thereafter flowers are allowed to develop for the fruit formation. So if you see in the diagram, so if you take a flower, in this flower we are using the forceps or using the scissor, we are removing the male part, that is anther. So removal of anther from this flower is called as emasculation. Following this emasculation, in order to protect this stigma from the unwanted pollen grain, there is a bagging required, and this bagging is done by this butter paper. And after bagging, when this stigma is reaching the receptive stage, this receptive stage where the stigma is able to capture, where the stigma is able to support the pollen tube germination, at that stage these desired pollen grains are brought together and they are dusted off on this stigma so that there is pollination, and then post pollination and fertilization events will go on to create the better varieties. So again after that this rebagging is again necessary to avoid the contamination. There is again another picture showing it's a bisexual flower having male and female both, and female is a stigma, pistil, this is entirely pistil, and in this stigma is usually is the stage where the pollen grains are landing. So if it is a bisexual flower, these anther are removed. So these anthers are removed. Removal of anther from this bisexual flower is emasculation, and then we are bagging it using the butter paper, and later on when it is receptive we are bringing the desired pollen grains and we are dusting off using this brush. So these desired pollen grains are dusted on this stigma, therefore this stigma is getting the desired pollen grain, and later on this stigma again is rebagged for the fruit development, fruit or seed development or any kind of development further which is desired. So in case of unisexual flowers, we have said that unisexual flowers are carrying only one reproductive part. It can be male or it can be female. So in such unisexual flowers, there is no need of emasculation. However, they need to be bagged because the stigma can take the unwanted pollen grain here. So it needs to be bagged using the butter paper. They are bagged, and once this stigma is receptive, again the same thing, the desired pollen grains are brought and these desired pollen grains are dusted off on this stigma, and this stigma further leads to pollen tube germination, and then this pollen tube will discharge its content to the fertilization event. So flowers are rebagged further to avoid the contamination. So this in case of male and this is in case of unisexual and bisexual. So here we have seen that how a desired pollen grain is brought together with the human intervention in case of artificial hybridization. So due to this artificial hybridization, there are so many crop varieties are developed here. So it is well followed and it is very much acceptable, and there is no modification of genes here. There is only selective breeding here. So this all about the pollination. We have studied about pollination broadly, and after this pollination, once these pollen grains they are discharging those contents through the pollen tube towards this micropyle where my male and female gametes are carrying, where the male and female gametes are coming together through this fertilization step.